So in today's seminar, I'll be talking about the IVF, which is the in vitro fertilization. So IVF, we all know, is one of the most effective forms of assisted reproductive technology. So it involves fertilizing an egg with sperm outside the human body in a laboratory dish and then transforming the resulting embryo into our uterus. So IVF is used to treat various fertility problems such as blocked fallopian tubes, male infertility, unexplained infertility and certain genetic disorders. So what are the steps involved in it? So basically the outline steps are, we do ovarian stimulation to stimulate the eggs, and then we retrieve those eggs, and then it goes for sperm retrieval will be done, and then it will be cultured in a dish. So embryo culture will happen, and then the cultured embryo will be transferred, and then there will be monitoring of the pregnancy which is going to happen. That is the luteal support and pregnancy testing. So this is the basic steps. So how these steps are performed is what we are going to see today. So ovarian stimulation, that is actually a controlled ovarian hyperstimulation. So the objective is to stimulate the ovaries to produce multiple mature eggs. So a natural cycle typically produces only one egg. So in IVF, multiple eggs are desired to increase the chances of successful fertilization. So we do hormonal medications such as follicle stimulating hormones and LH hormone, which are administered daily via injection for 18 to 14 days. So then the patient's response is monitored. We usually do ultrasound and blood test. So why ultrasound is because it measures the size and number of ovarian follicles, and blood test is to assess the estradiol, which is the E2 level, to evaluate the follicular activity which is taking place there. Next is the egg retrieval, which is the oocyte aspiration. So when follicles, the objective is to collect mature eggs from the ovaries. So when follicles reach the optimal size, which is 18 to 22 mm, a trigger shot of the human chorionic gonadotropin, which is the HCG, will be given. So to induce the final egg maturation, then the timing egg retrieval is scheduled 34 to 36 hours after the trigger shot of this HCG. Well, how this procedure is done is, it is done under the light anesthesia or sedation, and a transvaginal ultrasound guided needle is used to aspirate those follicular fluid and retrieve eggs. Eggs are then identified and collected by an embryologist in the lab. Like this egg is perfect to go for the next step. Next will be, we have collected the egg now. Now we have to move on to the sperm collection. So how to prepare and collect those sperm is the objective is to obtain and prepare healthy sperm for fertilization. So collection procedure involves masturbation on the same day as egg retrieval. So in case of male infertility, surgical sperm retrieval, that is TSE, testicular sperm extraction may be needed. Processing the sperm are washed to remove impurities and seminal fluid. The most motile and morphologically normal sperm are selected. Next is the fertilization process. We have collected the egg, we have collected the sperm, now we have to fertilize. So the fertilizer retrieve eggs with selected sperm in the laboratory. We use two main methods. First is the conventional IVF, where sperm and egg will be co-incubated in the same culture dish. So naturally the sperm will penetrate into the egg. So this is the conventional IVF. So what they are performing now is the ICSI, which is the intracytoplasmic sperm injection. A single sperm is directly injected into those mature egg using a fine needle. Especially this is used in case of severe male infertility. So after 60 to 20 hours, fertilization is checked by observing the presence of two pronuclei, which is one from the egg and one from the sperm. The next step after this will be the embryo culture. So this is done to monitor and support the early embryonic development. So fertilized eggs, which is now the zygote, are cultured into a controlled incubator. So after 3 to 5 days, they divide and develop into the embryos. I have talked about the cleavage phase and the blastocyst formation. So over 3 to 5 days, they divide and develop into the embryos. So now they will be assessed based on the cell number and symmetry, followed by fragmentation percentage. So progression to the blastocyst state, that is around day 5. Day 3 will be cleavage and day 5 will be blastocyst, which has the higher implantation potential. Next is the, there's one optional step here, which is the pre-implantation genetic testing (PGT), which can be performed to detect any genetic or chromosomal abnormalities, especially in older women or couple with known hereditary conditions. Next is the embryo transfer. So objective is to transfer selected embryos into the uterus, so which occurs on day 3 or day 5. So a soft thin catheter is inserted through the cervix into the uterus under USG. Next is the one or more embryos are carefully released into the uterine cavity there. Then the number of embryos transferred depends on the woman's age, embryo quality and their clinical history also. So we have transferred it, now we have to monitor the pregnancy. So there will be the luteal phase support and the pregnancy test. So this is to support the implantation and monitor for their pregnancy. So progesterone supplementation via vaginal gel, oral tablets or injections begins on the day of egg retrieval or embryo transfer. So progesterone prepares and maintains the uterine lining for the implantation. Next, the pregnancy test, a blood test for beta HCG is done 10 to 14 days after this embryo has been transferred. If positive, follow-up ultrasounds confirms the gestational sac and fetal heartbeat. So this is one of the basic things which we know, what are the steps involved. Now we'll concentrate on what are the latest technologies in IVF. So in current technologies which are done in IVF is, in recent years in vitro fertilization has witnessed significant technological advancements which are aimed at enhancing success rates, reducing patient burden and making treatments more accessible. So some of the notable developments include, first is the artificial intelligence in embryo selection and sperm analysis. So these AI algorithms are increasingly utilized to improve the embryo selection and assess the sperm quality, because these AI algorithms, they are extensively trained on those data sets where they have where they learn the patterns of the sperm motility, which sperm will be best. So there will be a large number of data sets for training, and so they will be able to identify which sperm is more fertile to get a higher chance of success rates in pregnancy, so higher number of implantation potential also. So it enhances sperm selection by evaluating their motility patterns and morphology, leading to better fertilization outcomes there. Next is the microfluidic technology for gamete selection. So microfluidic devices offer precise control over the microenvironment of gametes and embryos, mimicking the natural reproductive conditions. So these lab on a chip technology facilitate the selection of high quality sperm and eggs, improving their fertilization rates and embryo development. So this is one of the other technology which is combined with AI. Next is the automated sperm injection robots. So I think 2 days before there's one success of IVF using this automated sperm injection robots. So robots, innovations in robotics have led to the development of these AI operated machines capable of performing the intracytoplasmic sperm injection. So these systems automate the selection and injection of sperm into eggs, reducing human error and increasing the consistency in the IVF procedures. So yes, a recent case involved a baby conceived using this particular technology, which is being published in Nature Communications, I think so. I'll show the image of it during the end of this thing. Next is the alternative egg maturation techniques. So traditional IVF techniques, they require these hormone injections to stimulate the egg maturation and all. So which can be physically and financially taxing also. It is actually around 5,000 euros in abroad countries for this IVF convention. So this is usually a costly procedure. So a novel approach involves maturing eggs outside the body using ovarian support cells derived from the stem cells. So this method significantly reduces the hormone dosage which is given during the treatment duration, offering a less burdensome alternative for patients. Next, we also have advanced imaging technologies for the embryo assessment. So high resolution label-free imaging techniques such as the optical matrix imaging have been applied to embryology. So this OMI, which is the optical matrix imaging, compensates for the aberrations and scattering, providing three-dimensional high contrast images of the embryos at confocal resolution. So it helps us to get a detailed visualizations of the intracellular structures, aiding in the assessment of the embryo quality. Last is the in vitro gametogenesis, the IVG. So researchers are exploring this technique which involves creating gametes from somatic cells. So this approach could provide new avenues for individuals facing infertility, including same-sex couples to have genetically related children. So recent studies have demonstrated progress in this area also through though clinical applications remain in the experimental stage only. These advancements collectively aim to enhance the efficacy, safety and accessibility of the IVF treatments, offering a renewed hope to individuals and couples seeking the assisted reproductive solutions. So I'll just show the images of. So this is the normal IVF thing where they used to see. This is using the AI thing. What we able to see all the controls, how depth it has to go, how the needle has to penetrate, everything will be shown here. But this is only through normal thing. So this is the difference between of us using the technology for IVF thing. So this is from the article from I told about the baby conceived using this automated sperm injection. So this is how the automated sperm injection.
One baby.
One baby. For a 43-year-old.
No, there is also one more article. Like in that, I don't remember